since last summer, but I'll back up and say that I met Chau and got to know her in Biochemistry 1 class in the fall of 2019, you know, and a fall before we really knew what was coming. And so, so we ended the term, or ended the, 2019 year, moved into 2020, and next thing we know, we're all being sent home for spring term when she was taking Biochem 2. And like many students, that came with some disappointments, including a summer research program that was canceled for Chau, and study abroad, which seems so long ago, potentially studying abroad in Oxford in the fall of her senior year. But then she learned that she could come back to campus for the summer. And after a 2-week quarantine, we were able to get her in the lab. And really, she has been unstoppable ever since. So I think maybe have never had such a dedicated and driven student, work in my lab. She took on a project that was brand new and had a lot of ideas, but a lot of things that had still needed to be developed. And it was somewhat risky. There was plenty of opportunity for this project to fail altogether. But Chau has been diligent and dedicated and has done such high quality work that we're actually ending this at a place that's very promising. And so I'm so proud of her and all of her work, and am excited that she can tell you all about it today. So with that, I'll hand it over to Chau.
Thank you, Kim, for that very lovely introduction. Thank you everyone for being here. I will get my screen share started. Can you all see the screen? Okay, perfect. All right, so thank you for joining me in this virtual space. I am very excited today to be presenting on my work the past year in the Dixon lab. My talk today is titled Visualizing the Protein Angiogenin in Cancer Cells Using Split Fluorescence Technology. And before I start, I want to mention that all of my slides will be numbered. So if you have any questions, feel free to jot down the slide number and we can revisit them at the end. Okay, so let's get started. The premise of my research in the Dixon lab has been centered around cancer and the proteins that encode for cancer. So cancer is a devastating disease that is caused by the rapid growth of tumor tissues. And this rapid growth of tumor tissues is characterized by 2 biological events: the increased cellular proliferation and increased blood vessel development. Now, cancer cells are rapidly growing, dividing, and taking over healthy cells in our body, and that is exactly how tumors are able to spread so quickly. And to meet this increased demand in cell proliferation, we have more blood vessels gathering around these cancer cells. These blood vessels are supplying additional cellular resources for these cells to grow. And both of these biological events are heavily regulated by proteins, So one of the ways that we can better understand cancer is to study these proteins that heavily regulate these processes. Now, the Dixon lab is interested in studying a protein called angiogenin, or ANG for short. Here on the left, I have a crystal structure of this protein. If you squint at it, it kind of looks like a kidney bean. But for the rest of this presentation, I will be simplifying angiogenin with this Pac-Man shape molecule here on the right. Now, I think that this is a really cool protein, but that's because I've been studying it for the whole year. But here are some facts about the protein itself. It was first discovered in 1985 by a group of scientists isolating it from human cancer xenografts. The protein is encoded by 123 amino acids, totaling 14.4 kilodaltons in size. This protein is found in a variety of human cells, from endothelial to neuronal to cancer cells and more. In these cells, the angiogenin protein can act as a ribonuclease, where its main catalytic activity is to go after RNA and cleave them or cut them. Now, angiogenin is small but mighty. It can do many things in the cells, but for the purpose of our talk today centered around cancer, 2 of the most important functions of angiogenin is that it is very good at forming new blood vessels, and also very good at upregulating cell growth. So let's first look at the first function, the growth of blood vessels. Now, blood vessels are made up of endothelial cells, which are these blue cells in the middle of the screen here. These endothelial cells on their surface have a protein called actin. angiogenin can actually come bind to this actin protein, and that binding will help this whole cell dissociate from that tissue and migrate to neighboring spaces. And the cellular migration behavior, as well as downstream processes, is exactly how new blood vessels are able to be generated. This process is actually formally referred to as angiogenesis, which makes a lot of sense because the protein that encodes for it is angiogenin. So now moving on to the second function of angiogenin in cellular proliferation. In order to perform this activity, angiogenin needs to get into the cells. It can do that by interacting with a cell surface receptor, as shown in purple here. Now, once the protein is inside the nucleus, it can go after RNA and perform its catalytic activity, i.e. cleaving RNA. Now, angiogenin is very specific about what kind of RNA it can cut, and one of the example substrate is non-coding RNA that actually need to be cleaved for ribosome production. So after that RNA is cleaved by angiogenin, we get more ribosomes, which in turn allow for more protein translation, including the proteins that encode for cell proliferation. And this is exactly how angiogenin can contribute to the whole cellular proliferation process. So now knowing these two functions of angiogenin, the kicker is that angiogenin is upregulated in tumor tissues, inside the cell nucleus of the cancer cells, as well as in the extracellular space surrounding endothelial cells. And this in turn leads to both increased cellular proliferation as well as increased blood vessel development. Now you might be thinking, can we find a way to tune down this angiogenin upregulation and target cancer that way? The short answer is yes, but the long answer is we need to know more about how angiogenin works in the cells. Particularly, one of the most pressing questions for us is how exactly does the cellular entry of angiogenin change when the cells are experiencing stress? Is this process any different from healthy cells versus like cancer cells when they are experiencing a lot of stress? And this is exactly where my research comes in, trying to better develop a system to better understand this process. I want to make a note here that in this presentation, I will be using three terms interchangeably: entry, internalization, and uptake, all referring to the activity of angiogenin getting into the cells. So now the next task that we need to deal with is that most proteins are invisible. So we need to find a way to see these angiogenin protein to study its uptake. And with that, thankfully, we have another group of proteins that are not invisible. These are called fluorescent proteins. Now, as you might have heard or know or guess, these fluorescent proteins can fluoresce, meaning they can give off light at a certain wavelength. One of the most commonly used fluorescent proteins in biological research nowadays is green fluorescent protein, or GFP for short. Now, GFP was first discovered in a jellyfish species that can glow green, really cool stuff. And this is why I started my presentation with a jellyfish picture, if you noticed that. Now, GFP can give off green fluorescence because it has a chromophore, which is this orange molecule in the middle here. Here on the right, that's what the chromophore looks like. This chromophore can actually absorb light in the blue light wavelength and then emits light in the green light wavelength. So blue light around 480 to 490 nanometers and green light in the 500 to 530 nanometers. And this green emission is exactly how GFP can fluoresce. Now, in biological research, GFP is often used as a reporter molecule where it is tagged to the protein that you want to study. So in our case, that'll be the invisible angiogenin protein. This gives us a fusion protein complex that will glow green. So here I have a fluorescent microscopy image where the cytoskeleton of the cell has been labeled with GFP. And as you can see, it is very clear for us to visualize the cytoskeleton of the cells using GFP. This is really cool, but it gets even better. So biologists are really clever people, and they have been able to engineer GFP to make full use of the system. And split fluorescence or split GFP is one of those ways. And before I explain split GFP, I want to take a deeper look into the structure of GFP itself. So you might have already noticed that GFP has this really cylindrical, symmetrical structure that resembles a barrel. This barrel is made up of 11 beta strands, which are these long green strips kind of crisscrossing over on the outside of the GFP molecule. This barrel is actually very critical to maintaining the fluorescent property of GFP because it can protect the chromophore that is in the middle. So once you break apart this GFP structure, GFP is actually not fluorescent anymore. And that exactly is the working principle of split GFP. Now in split GFP, the 11th strand of the beta barrel is separated from the rest of the molecule, giving us two non-fluorescent molecules, GFP 1 to 10 containing the 1st to the 10th strand of the barrel, and GFP 11, which is the 11th strand of the barrel. Now we can tag the GFP 11 to the protein that we want to study, giving us a smaller fusion protein that would not be fluorescent if it is by itself. However, once you combine it with the GFP 1 to 10, the other fragment, these two can spontaneously reassemble into the full GFP barrel, and we end up with a fluorescent fusion protein. Now here I have another depiction of this system, and there are multiple advantages to this system. First of all, it is very sensitive. What that means is the GFP 11 and the GFP 1 to 10 by themselves are not fluorescent. So we would expect to see virtually no background fluorescence. However, once they are able to combine, we get fluorescence. So fluorescent is an indication of the successful reconstitution between these two fragments. Now, secondly, the GFP 11 tag is very small. It's only about 1/10 of the size compared to the full GFP molecule. So this tagging is very non-invasive and versatile because the GFP 11 tag will not disrupt so much of the protein that you want to study. So now keeping in mind these very cool advantages and the question that we asked earlier about angiogenin uptake, this is the time for us to put the pieces together. My research focused on developing a reporting system for angiogenin using the split GFP technology. And we hope to use this system to understand angiogenin uptake in cancer cells. Here's how it works. I first created fusion proteins with angiogenin and GFP11, as you can see here on the left. I then introduced these fusion proteins to a human cell line, target cancer cell line that is HeLa cells. This cell line has been engineered to express the non-fluorescent GFP 1 to 10 component, as you can see in gray here. So now when the fusion protein enters the cells, GFP11 can bind to GFP 1 to 10 and fluoresce. So we can, in other words, fluorescent is an indication of the successful cellular entry of angiogenin fusion protein. Now, our fusion proteins need to achieve 3 main features for the system to be fully functional. First of all, they need to have conserved structure and catalytic activity. And to be more specific, we will be testing for the activity of angiogenin coding RNA. Secondly, these fusion proteins need to be able to restore GFP fluorescence. And lastly, they need to be able to enter live HeLa cells that we want to study them in. Okay, so with these 3 features, let's first start with the first step, making the fusion protein with the correct structure and activity. Now, here is a simplified diagram of the process of protein expression and purification. I want to direct your attention to the left side of my screen here, where we will be talking about recombinant cloning. And this essentially is the process of making the DNA sequence encoding for my fusion protein of interest. And before I go into more depth about the exact fusion protein design, I want to give a refresher on the primary structure of proteins. So proteins are made up of amino acids, as you can see in these little colorful circles here. This makes up a long peptide chain that has 2 ends, the C terminus and the N terminus. And being able to distinguish between these 2 termini is important to understanding the design of my fusion proteins. And I do want to say that in fusion proteins in literature, usually the GFP part is attached to the C terminus of the protein that you want to study. Okay, so keeping that in mind, here is a representation of my angiogenin protein in red, with the N terminus on the left and the C terminus on the right. I first experimented with tagging GFP11 on the N terminus of angiogenin. This gives me N-terminal tagged protein, or NXAC, where the X stands for the number of GFP11 tags. So as you look down this column, you can see that I experimented with 1, 2, or 4 GFP11 tags. In theory, the 4x GFP11 tag would give us 4 times fluorescence compared to the 1x tag, which is very promising. Similarly, I also experimented with the C-terminal tagging, where GFP11 is tagged on the C terminus of angiogenin, giving me another 3 C-terminal tag proteins, or NAXC, where the X is the number of GFP11 tags. In total, I have 6 fusion proteins. Now, you might think this is a lot, but any molecular biologist can tell you that cloning is not fun. And fusion proteins are very unpredictable. So with a suite of 6 fusion proteins, I was hoping to identify at least 1 fusion protein that satisfies the 3 criteria, as I mentioned before. Okay, so now after I have the DNA sequence encoding for my fusion protein, I introduce them into a plasmid through a process called Gibson assembly, and I can go into more depth about that process if you're interested later on. But with this plasmid, I then introduce them to E. coli cells, which is a bacteria that is very good at making large amount of protein in a short amount of time. These E. coli cell lysates are then carried through a purification process, giving me the pure fusion protein at the end. Okay, so one of the ways that we can make sure that I made the right protein and this protein is pure is to analyze them using SDSPAGE. SDS stands for sodium dodecyl sulfate, which is a detergent, and polyacrylamide gel electrophoresis is the long name for PAGE, which is a way for us to study these proteins based on size, or sort these proteins based on size. Now, with my fusion protein, I first need to denature the protein using SDS. This disrupts the tertiary structure of the protein, turning it into a long linear peptide chain with negative charges all embedded all throughout the protein. I then load this protein onto a running acrylamide gel, connect it to a power supply, and now all of my negative charged proteins will, with this power supply, will migrate down the gel towards the positive electrode at the end, at the bottom here. And this running behavior will help sort the proteins based on size, such that the small fusion protein will end up at the bottom and the large fusion protein will end up at the top. Now, I like to think of this as running through a crowd. If you're tiny, like my baby sister, you can run through a lot faster than I can, like a full-size adult. Probably not the best analogy, but you get the idea. It is also for us in this experiment to include a protein ladder, which includes proteins of predetermined sizes, and this can help us find out the size of our protein of interest. So here is a picture of my SDSPAGE gel. This is of the NA1C protein, which is the 1xGFP11 tag on the C terminus of angiogenin. On the left here, I have a sample protein ladder with predetermined sizes in kilodaltons. So here's how you read this gel. This first lane includes E. coli cell lysates without the protein, just happily growing. The second lane includes E. coli cell lysates with the protein after induction, as you can see in this big band here. This is then carried through a purification process, giving me a pure fraction of protein at the end of the right size, which is 17 kilodaltons. Now, similarly, I was able to express and purify the 2x and the 4x fusion proteins, where the 2x fusion protein had a size of 19 kilodaltons and the 4x fusion protein had a size of 23 kilodaltons. So in total, I was able to make 3 C-terminal fusion proteins of the right size, and I will be proceeding through my talk today, focusing on these 3 fusion proteins. Okay, next task for us is to make sure that these fusion proteins are active. And to be more specific, we are testing for the activity of angiogenin to cleave RNA. So with that, I'm using a fluorogenic RNA substrate. This molecule includes a reporter molecule R, a quencher molecule Q that is connected by a short length of RNA in the middle. Now, the reporter molecule can receive blue light, emits green light, which is in turn reabsorbed by the quencher molecule if the RNA is intact. This gives us no signal. However, once you introduce angiogenin or any other RNA cleaving enzymes, the reporter molecule can now diffuse away from the quencher molecule and fluoresce green. So this green fluorescence is exactly what we hope to see on the plate reader, where fluorescent will tell us that our angiogenin portion of the fusion protein is active. So keeping this in mind, here is my graph, my data from this experiment. On the y-axis, I have fluorescence in arbitrary units, and on the x-axis, I have time in minutes. So in this experiment, I included wild-type angiogenin in red, which is angiogenin without any modification. And as you can see, angiogenin is actively cleaving that substrate, and we get fluorescence, increasing fluorescence over time. So similarly, for our 3 fusion proteins in green, we also see that there is an increasing level of fluorescence. Now, this goes at a slower rate than wild-type angiogenin, which is not ideal, but this is enough for us to kind of suggest that these fusion proteins folded properly into the correct structure in order to give good RNA cleaving activity. Okay, so coming back to this checklist, we are able to check off the first box. We made the right fusion protein with good structure and activity. So the next step is to ensure that these fusion proteins can restore GFP fluorescence. As a reminder, here is what we are looking for: GFP11 binding to GFP 1 to 10, giving off green fluorescence. So for this, I harvested my target cells, so HeLa cells GFP 1 to 10, lyse them, meaning breaking them open, centrifuging to remove any soluble components, which eventually gives me a clarified cell lysate containing the non-fluorescent GFP 1 to 10. I added this to a microplate along with my fusion protein, incubated the plate at 37 degrees for half an hour, then let it cool to room temperature before doing fluorescence reading. So once again, we would expect to see green fluorescence, where green fluorescence tells us that GFP11 bound to GFP 1 to 10 and restore the fluorescence of GFP. Okay, for data, here is my graph from that experiment. On the y-axis, I have fluorescence in arbitrary units, and on the x-axis, I have 5 different treatment groups. So I included 2 negative controls where the lysate was added with water or wild-type angiogenin. Now, both of these, we expect to see a low level of fluorescence because there is no GFP component to restore GFP signal. And that is exactly what we see here. In contrast, for our fusion proteins as shown in green, we see a 3-fold increase in fluorescence. And this tells us that the GFP11 component on the fusion protein did bind to GFP 1 to 10, restoring fluorescence. So coming back to this checklist, we are able to check off the second box as well. Our fusion proteins were able to restore GFP fluorescence. So our last step is to demonstrate that these fusion proteins can enter live cells. So for this, I want to give a little bit more background to help us understand the activity of angiogenin getting into the cells. Here is a crystal structure of angiogenin, once again in gray. Angiogenin actually has a receptor binding site, as I highlighted in blue here. And this receptor binding site is exactly what helps angiogenin interact with the cell receptor to get into the cells. Now, if we want our fusion protein to be able to enter the cells, we want to make sure that this binding site is accessible to the receptor. Here I have some comparison of the crystal structure of wild-type angiogenin on the left and the fusion proteins in the middle on the right. The receptor binding site is colored in blue and the GFP11 is colored in green. There is a trade-off in these fusion proteins for the size of the GFP11 and the cellular entry capacity. The 1x GFP11, it is pretty small, so it's probably not going to interfere with the whole fusion protein from getting into the cells, but we would get a low level of fluorescence compared to the 4x GFP11, where we get theoretically 4 times as bright of fluorescence. However, this bulky tag might very well hinder the protein from getting into the cells. So keeping this in mind, here is my experimental methods for this experiment. I introduced 5 micromolar of my fusion protein onto live HeLa cells. So emphasis on the live here, because the last experiment was on broken apart cells. Now these cells with the protein will be incubated at 37 degrees under standard tissue culture conditions for 2 hours, and then they are studied using flow cytometry. So flow cytometer is an instrument that allows us to sort cells based into single cells and then study their characteristics using fluorescence. In our case, we want to hit these cells with the laser beam at 490 nanometers excitation wavelengths, and then record the level of fluorescence at 530 nanometers. All of this data is collected into a computer, which turns out two different kinds of plot, a dot plot and a histogram. A dot plot can help us distinguish the live cells from the dead cells, and within the live cell population, we can then study the level of fluorescence in that live cells using histogram. Now I'm going to show you a couple of sample pieces of data to help us reorient ourselves and see what we should expect. Here on the left, I have a histogram with fluorescence on the x-axis and the cell count on the y-axis. For our negative control cells, so cells without the fusion protein, we would expect to see a low level of fluorescence, say about 20 units. Now, this mean in the histogram, the mean fluorescence in this histogram is translated to a bar graph on the right-hand side, where fluorescent is now on the y-axis, excuse me. In contrast, for the experimental group where a fusion protein is introduced to the cells, this will give us a high level of fluorescence, say about 50 units. This is in turn translated to a bar graph on the right. And as you can see, looking at this bar graph, there is a very clear difference in fluorescence between the negative control and the experimental group with the fusion protein. And that is exactly what we hope to see. Okay, so first I wanted to experiment with the largest fusion protein because, as you might recall, this has 4 of the GFP11 tags, theoretically giving us 4 times the level of fluorescence. Now here's my data. I have my histogram on the left, my bar graph on the right, with the negative control cells in gray and the fusion protein experimental group in green. As you can see, disappointingly, there is no statistically significant difference in fluorescence signal between the negative control group and the experimental group. And this suggests to us that the largest fusion protein did not enter the cells, possibly because of the bulky GFP11 tags. So now, next we wanted to turn to the smallest fusion protein. Maybe this will enter the cells. Now here is my data. I have my experimental group in green again and my negative control group in gray. Now, these cells are also showing no statistically significant difference in fluorescence signal. Actually, the fusion protein is showing a lower level of fluorescence, which might very well be within the error of the instrument, and I will be explaining that a little bit more later on. However, taken together, this suggests to us that the fusion protein did not enter the cells either, disappointingly so. Okay, now there were a couple of issues with this experiment. First of all, we were skeptical that our instrument, the flow cytometer, is not in its best condition. We did a positive control experiment that failed to show the level of fluorescence that we would expect to see. And then from that, our first line of hypothesis for why this failed is that maybe the fusion protein did enter the cells, but the fluorescence was not picked up by the flow cytometer. And this could be verified with a couple of different experiments, as I will mention in a little bit. But another line of hypothesis is also possible, where the GFP11 tags have hindered these fusion proteins from getting into the cells. Now, if you look at these crystal structure again, the GFP11 tags are on the same side as the receptor binding site in blue here. So it is very well possible that these GFP11s are obstructing the binding site from binding to the receptor and therefore precluding these fusion proteins from entering the cells. So here are a couple of ways that we can validate this uptake. Number one, easy solution, go find a better flow cytometer and calibrate it properly before use. Secondly, probably something that is more doable here at Lawrence, we can attempt immunocytochemistry. This is an antibody-based experiment where we can label the angiogenin portion of the fusion protein in the cells using primary antibody and then tag on secondary antibody that can give off fluorescence. So any signal from the fluorophore can tell us that our fusion protein did enter the cells, but which is not detected by the flow cytometer. So now I know that it'll be really nice to end on a definitive conclusion, but I'm still regardless very excited about where this project is headed in the future because it will be a very powerful system. And coming back to this checklist again, we have not been able to demonstrate the capacity of these live cells the capacity of this fusion protein to enter the live cells, and more data collection shall be continued by future research students. But once the system is optimized, here are a couple of ways that could be very useful. First of all, it allows us to use fluorescence to monitor the uptake of angiogenin in real time. And what I mean by that is we hope to functionalize the system for use in microscopy tools such as confocal microscopy down the line in order to study uptake in live cells. Emphasis on the live cells because in the past, immunocytochemistry is usually the technique that researchers have used to study angiogenin uptake, and that only allows us a one-point endpoint data point for fixed cells, basically like dead cells. So secondly, this also allows us to study angiogenin uptake in other cell lines because the system is very adaptable. All you need to do is to get the cell line that you're interested in studying, engineer them so that they express the GFP 1 to 10 component, and then voila, we have another system to study angiogenin in those other cell lines of interest. Now, zooming out a little bit, this system also will allow us to work towards elucidating the full mechanism of angiogenin in the human body. And with that, we can hopefully identify whether angiogenin is a good therapeutic target. And if it is, we can develop more selective angiogenin targeting therapies. What that means is we want to target angiogenin in only the problematic cells, like cancer cells, and not interfering with the activity of angiogenin in healthy cells. And with that, I would like to wrap up with some acknowledgements. I would not have been able to complete this research with so much encouragement from many of you. First of all, I'd like to thank Kim, my research advisor. Thank you for being an amazing research advisor in the past year and always believing in me. I've learned so much in the past year. I also want to take this space to acknowledge the previous students that have worked in your lab, particularly Sophie, who have set the foundation of this work and set really great examples for me to follow. I want to thank the chemistry and biology faculty, Stefan, Allison, Mike, Graham, Deanna, Rachel, Shannon, all of you have taught me chemistry and biology, biochemistry, and for making me love this field that I'm pursuing. I want to thank the biology stockroom with a special shout out to Eric and Megan for being my lifeline when things go south in the lab. Without you, I probably would not have been able to complete this work here today. I want to give a special thanks to my classmates and friends here in the US and also around the globe. Thank you for being really supportive. And special shout out to my capstone group for helping me revise this talk. So that's Erin, Jojo, and Lindsay. I want to thank my family, my parents, my two little sisters, one of whom drew this very cute picture of me that I want to share. Thank you to my extended family for always having my back and believing in me, although we are very many miles apart. And last but not least, I want to thank all of you, 55 of you, wow, who are joining me from either the US or anywhere around the world, Vietnam, Europe, Australia. I really appreciate all of you. Thank you for being here. And with that, I will take some questions. Here is a recap slide to help you jog your memory as needed. Thank you.
Congratulations, Chau. That was fantastic.
Thank you.
I'll mute so people can ask questions.
Yes, Megan.
So I just had a question. You did most of your work with like the C terminus GFP. What happened with the N terminus GFP?
That is a great question. I actually spent about half a term trying to express my N terminus protein, and that did not work in the current E. coli BL21D3 system that we have. So we decided to put that aside and focus on the C terminal proteins first. And I do want to, I did mention this in the presentation, but in literature, it's usually more common for people to tag the fusion protein, the tag GFP to the C terminus, because that's the end that gets turned out after the actual protein that you want to study during translation. So that is for future research students to do.
Cool.
Graham.
Yeah, Joe, thank you. Great talk. A question I'm a little curious about, and this might just be my lack of biochemical knowledge. Why would you expect the 4 times tagged proteins to elicit more fluorescent response? Because you only have one G1 through 10 receptor for one kind of tag to fit into. So why would you expect more fluorescent response with more of those tags?
Yeah, that's a great question. So actually in my protein design, each of these GFP molecules are spaced out by a linker molecule. And this is a design that has been optimized in the Wiseman lab at UCSF. So this, essentially what that linker does is separating the GFP11s far enough so that each of them can associate with the GFP 1 to 10. So theoretically, 4 of these GFP 1 to 10 can bind to GFP 11 and therefore give us 4 times the level of fluorescence in each fusion protein. Does that help?
Absolutely, thank you.
Eric?
Thank you. This is just beautiful work. I kind of have a follow-up question about the GFP11 tagging. When you were looking at the ability of that GFP11 in the cell culture lysate to generate fluorescence, did you do any kind of dilutions of that to see whether or not you might be limiting for the cell culture lysate GFP 1 through 10? And do you think that you might be able to get higher fluorescence out of the multiple GFP tags than you did out of the single if you were to do that?
Yeah, that is a great question. I do have a backup slide where I can show you the results from that experiment. Just give me 1 second. Oops, this is the one. So I did do dilution of my HeLa-cell lysate. And here on the x-axis, you can see that different amount of GFP lysate was added. We do think that the limiting factor here is the lysate. So the more lysate you add, the higher the level of fluorescence that you can see. Sorry, did I answer that question?
Yeah, that looks really neat. I wonder if even at the top ratio of lysate to Na4C, if you might still be limiting. So that looks like a really cool step in that direction, because it seems like things are starting to spread out a little bit in the way you would expect.
Yeah, for sure. I do think that this could be very well limiting. This lysate could be very well limiting because in this experiment, I added the fusion protein in a 2-to-1 ratio to the cell lysate. And that was just my way of making sure that the protein is not the limiting factor, but it seemed like it had swayed the other way. So future research students, that's what you should explore. Shannon?
Yeah, really elegant experiments and really beautiful PowerPoint slides. I want to have your PowerPoint skills.
Fire render.
So I guess I had 2 questions about like future experiments that you could maybe do. So I know one of your kind of concerns or things that you wanted to troubleshoot was about why maybe it was internalized into the cells, but we couldn't detect the fluorescence. Have you thought about doing just endogenous expression of the GFP11 tagged angiogenin, and would that endogenously express in the cells, and would you be able to detect it from that kind of?
Yeah, that's a great question. That was one of my questions asking Kim too, like, why can't we just do that? And I think there are 2 reasons to that. The first is more of a logistical reason, like it's much harder to do transfection in a human cell line compared to a bacterial expression system. And the second reason is angiogenin itself is a secreted protein, meaning it will be secreted and uptaken by these target cells anyways. So it doesn't make as much sense to like make the protein within the cells because it will be exported and then re-imported.
Okay, could you mutate the exporter? I don't know. I don't work with human cells, so now I'm like, that might be very hard for bacteria to just like knock it out.
Yeah, I also have not had any experience with a human expression system, but that is a question that I had as well. So.
Okay, and then my other question is, are there any ways that you could do kind of in vitro interactions between the proteins the receptor and the angiogenin protein. So if the question is like, is it not being taken up because it's not interacting with that receptor? Is there a way that you could address that question in vitro?
Yeah, that's a
doing
great
those
question.
kinds of assays?
Yeah, I know that the receptor was recently identified. This has been very elusive. So angiogenin has been around for like 30 years, but the identity of the receptor was only identified in like 2017. So not much is known about it yet, but I think there's probably a way for us to do like cross immunostaining where you have maybe like the receptor on a plate and then add your my fusion protein and see if there's any cross-linking between the two.
Yeah, awesome. Well, great project. Thanks so much.
Thank you. Thank you. Any other questions? Yes, Eric.
One more. This is just a curiosity question. How does it get from the presumably, does it go in through endocytosis to the cell initially?
Yes.
Does anybody know how it gets across the membrane from the endosome into the cytosol?
I have not looked into that personally, but I know that angiogenin is kind of unique in the fact that it does not like to hang out in the cytosol a lot. That once it gets into the cytosol, it kind of is redirected to the nucleus straight away with a nuclear localization signal. And that is important to all of the cellular proliferation activity. But to your question, I don't exactly know how the endosome is broken apart. So angiogenin can hang around in the cell. I don't know, Kim, if you know, feel free to jump in there.
Yeah, no, but there really isn't anything known about the membrane crossing part of things. The where, when, how, not too much known.
Cool.
Yeah.
Thank you.
Well, if there aren't any other questions, I would also like to thank everyone for coming today and helping to celebrate all of Chau's impressive work. And I see that Rachel is host now, so I don't know if Rachel, you have announcements to make too. But before we end things, let's definitely give Chau another huge round of applause. Yay! That was
Thank you
fantastic.
everyone for being here. Now I can't stop sharing.
All right. I guess this wraps it up. Graham, do you have any